reference gene, what happens is that since it's a real time, it's a very sensitive approach. So even if there is a minor error, and that can lead to a dramatic change in the gene expression values. And what could be this minor error? This minor error could be any variation in the quality and the quantity of the RNA, because each and every extraction procedure or the purification procedure will have different efficiency, and that can lead to differences in the RNA quality. If you are not estimating correctly the RNA quantification, then there is also a chance that for the cDNA synthesis, you will take a different quantity of the initial RNA sample. That can lead to change in the copy number of the gene in particular sample. Also, we need to consider that the nucleic acids are unstable, so there are chances sometimes that if you are keeping it in room temperature, or if by mistake if something has happened to the cold storage or deep fridge, then there is a chance then that the nucleic acid becomes unstable or it's degraded. In some cases, it is also not an assurance that how you took the initial sample. One of the example is that suppose you took 1 ml of the blood of any HIV patient, and so the patient can have different number of cells per ml of the blood depending on the disease stages, right? So it means that even though you took initial sample size as 1 ml, but there are chances that the number of cells vary. Similarly, suppose if you are working on an algal tissue or the plant tissue and you weigh 100 milligram as an initial sample for extracting RNA, there are chances that you can get either 1 or 5 milligram, or you can take 95 milligram. So those kind of small variations can lead to changes in the gene expression value. Also, because of the poor primer selection, or sometimes what happens is that even if you have done everything correct, there are chances that you can do a pipetting error, or that can lead to the variation in the amount of the sample you are introducing into your reaction, or the template you are introducing into your reaction. Most of the time for the real time or for this kind of assays, we are using 0.5 microliter or 0.2 microliter, and even if there is a slight variation, that can lead to a dramatic change. So that is why we need a reference gene. So what this reference gene means? Reference gene means something which assures that there is no problem in the technical problem in your analysis, and whatever the differences in the expression is meant, that is due to the biological activity or the experiment what we are performing because of that. So one particular example which I would like to give here is in the figure, the red figure here, you see that you have a gene of interest which is expressed in A, B, and C three conditions, and then you have you can see that in A and B it is relatively similar, whereas in C you have a higher expression of your gene of interest. So that can be because that there is a genuinely sample C is having the high expression, or the second chance is that by mistake or by pipetting or for any other reason which I have discussed just now, because of that there is a more template in the C sample, and that's why it is showing you high expression. How you can overrule that possibility is by using a control gene. In this case, let us suppose if we use a tubulin, which is a housekeeping gene, and then that you can see that it has expressed in the equal amount in equal number in all the three conditions. So it can assure that you have done everything correctly up to the best of what you can, and the differences what you are seeing in your gene of interest in the condition C is a valid increase in the gene expression. Now consider the second example in the green box over here. You have A, B, which is increasingly expressed in comparison to sample C. Now it is possible that A and B you have taken more tissue or more RNA or cDNA or initial template, and less in C. Or it is also possible that this is correct. How will you identify it? When you have a control gene, let us suppose again tubulin in this case, and you can see that A tubulin, which you thought that it will be similarly expressed in the condition to B, is highly expressed in A. So it means that your control is also high. So there is a chance that by mistake or by whatsoever reason, you have included more template in the sample A. And this gene expression is not a result of your experiment between A and B. It may be simply a technical error at your end. So that is also possible. Now, most of the commonly used reference genes are the housekeeping gene. For example, 18 srRNA, 28 srRNA. In case of plants, we are having this or in general, we have a lot of cytoskeleton related genes like actins, tubulins, and then we also have the cyclophilin and elongation factor, GPDH, and so on. So mostly these are the housekeeping genes. Now, if we are relying this much upon our control, or you can say a reference gene for the gene expression data, then the reference gene itself should be of a very good quality. Now, if we are blindly using this housekeeping gene as a reference gene, and if this housekeeping gene itself is expressed in the experimental condition what we are studying, then basically you are making a mistake. So initially this was not realized, and what it was thought that most of the time people did that commonly based on the literature survey, they used actin and tubulin in most of the cases, which later it started, people started questioning based on the variation in the gene expression of these housekeeping genes. So, as I was saying, that there are several factors which can affect the expression of the housekeeping genes as well. Kind of the tissue which you are taking, the developmental stages of the organism which you are studying, whether there is a role of this housekeeping gene in fighting against abiotic and biotic stress, or during the diseases, infection, tumor, or if the gene is having some other properties like alternate splicing. So all these factors affect the gene expression of housekeeping gene. So it is not always good, or it is not at all advisable that blindly you take any housekeeping gene as a reference gene and that you use for your gene expression study. So which genes can be used for reliable reference? Well, for those genes, there is a constant expression between the condition which you are studying. There should be a minimum variability among the tissues. They should have the similar CT value to the gene of interest, and it should not have polymorphism or alternate splicing. Now, nowadays, what is happening is that for each experimental setup what you are setting up, you have to define your own reference gene based on the condition what you are studying. As for example, if you are studying a non-model organism, and if you are treating your organism to unconventional stress sources, as for example decrease in the pH or increase in the carbon dioxide, or any such conditions which is not previously studied in much greater detail, you may have to use at least 8 or 10 different genes. Again, common housekeeping gene, as well as some of the genes which you think that should not be expressed much in your experimental condition. You have to take all those things, design a primer, do the real-time PCR in triplicate, and then find out that which can be a good reference gene for your case. So the overall concept for the reference gene is that it should not reflect variations because of handling, and it should be not only changes in the expression due to handling, but also understand in this way that whatever the variation what you are observing in the two different conditions in this housekeeping gene, it should not be because of the handling error or technical error, and it should be mostly because of the biological variation within the sample. convenient among the different samples. So to do that, when this the problem of reference gene was emerged in the early 2000, because the qPCR was in use since 1992. However, till 1995 or 1996, people have not realized the importance of the reference gene. After that, when they realized that there is a possibility that one can do the gene expression study in a wrong manner, there are several people who came up with the different softwares which predicts the stability of the gene. Three of the major software which is widely used in the gene expression studies are Genome, BestKeeper, and Norm Finder. Now, what Genome does is that it identifies or it makes a stability index which is dependent on the average variation among pair of genes by comparing the control gene and with the other validated candidates. So basically, how it defines the gene stability is by measuring the standard deviation of the logarithmic transformed expression values. So for each gene in the different conditions, it defines its expression value, converts into the logarithmic scale, and then compare it with each other. And that gives them which is more stable and which is less stable. So based on the ranking. The second one was the best keeper. Here more statistical analysis were done, mostly geometric mean, arithmetic mean of the CT values of the gene in the different conditions, and also observe the standard deviation and the coefficient of variations of the CT values of each and every gene, and afterward it was compared with all. In the Norm Finder, which is again a highly cited software for finding the reference gene, it is also based on stability, but it also considers intra and intergroup variation. So basically, not only between the experiment and the control, but within control and within experiment also, those values are also taken into consideration, and based on which it defines the stability values. So these three softwares are majorly used for the reference gene finding for last almost 20 years. Now, the software which we are going to perform is RefFinder. It integrates the three softwares, the values of the outcome of the three software, and also the comparative delta CT values. So after integrating the four methods, it compares and rank the tested candidate referencing. So what it does is that it makes the work very easy, because in one click you will be able to get the result for all the different known software for the referencing finding. So if you can see this green box, you have the genome which is dependent on the stability index, that is the M value. Norm Finder, which is also depending on the stability value but considers the inter and the intra-group variation. And then the third, you have the best keeper, which is largely dependent on the coefficient of variation and the standard deviation of the CT values of different genes. Okay? Now, let us go and do some practicals. So you also have got an Excel sheet labeled as reference gene, and you will see that you have two conditions here. That is a controlled condition, and then you have the stress conditions. So this is just a customized data which I have for the analysis. Right? So you will see that I have taken some of the common genes which are used for the reference gene. Actin, then you have this ubiquitin enzyme, you have cyclin, tubulin, 16S, another gene for ubiquitin, then you have elongation factor, another ubiquitin, and then you have the butyric triphosphate. So just a random combination of 1, 2, 3, 4, 5, 6, 7, 8, 9. A random combination of this 9 gene, right, in two conditions called as control and the stress. In the second link, I mean in 5, you might have got a link which is a link for Refinder. Open your browser and you copy it. So it will take you to a link where the Refinder is hosted. You can read about it. So it integrates the genome, norm finder, best keeper, and delta city method. You can input your data. So what you need to take care is you can check the example how it is. However, I have already formatted the data how it wants. It wants in a way that you have gene name and then you have the expression value. You can also go and check that how the example is. So you have the gene name and then the city values. So similarly, you can copy it and then you can paste your data, analyze. Right. So what it does is that it calculated the delta city, best keeper, norm finder, and genome value, and then it gave you the ranking order from 1 to 9, and based on the two method, that is delta city and the norm finder, we have 16s as a good gene based on the data which we have provided. The second is between, so what it does is that it gives you a comprehensive ranking based on all the four methods. Now, if you will see that the 16s is undisputed, and I mean, in two of the software, 16s is the best gene based on the stability. So it is giving you first rank. The second rank is EF1 because you have here among the top 1, 2, 3, you have 2 times the CF1. Similarly, you have tubulin here and here. And then you have UCE1 because UCE1 was selected in the second one, right? So based on that, it gives you a it recommends you to the comprehensive ranking. You can again go back 1 to 1 and then look how the data looks. First is the delta city value. So what it plots here is that average of the standard deviation of the delta city value of 16S RNA gene. It means that within the experimental and the control condition, what is the delta city value and then what is the standard deviation of that average. So based on that, it calculates the most stable gene in the first part and the least stable gene in the second part. Similarly, for the best keeper, as I said, that it also it is very much dependent on R square value, that is the correlation coefficient, and the so best keeper takes into account the standard deviation and coefficient of variation of the CT values, right? So in this one, you can see that it has calculated the Pearson correlation coefficient of the best keeper, and then based on the gene, it has given you the most stable gene and the least stable gene, and according to which EF1 is the most stable gene in this case. The third one is the norm finder, which is also depending on the which is also calculating the stability of the gene based on the inter and the intra variation of all the conditions, and based on that 16S is the best gene for it means that it is the highly stable gene, and cycling is the least stable gene, right? And with the genome again, this is also calculating the stability value, and you will see that tubulin or UCE is having the similar stability value and ranks first. Now the choice. So you have seen that different method uses different algorithms or different calculations to calculate the stability value and gives you a different result. How would you now decide that which one you should go with? So in this particular example, if you are keen to go just for one reference gene, it is better that you go with 16S RNA gene because it has been predicted by two of the softwares. However, these days people are going with the multiple reference gene, and in that case, you can choose actually all the three 16S, EF1, and tubulin. If you have to conduct the experiment for few number of times with a limited number of genes, then it is advisable always to go with the three reference gene. However, suppose if you are limited with the cyber green or the other laboratory facilities, or let us suppose fund wise, then you have to take a call, and then you can go either with 16SR or 16SR EF. So the choice is completely yours because the software has done its work, and it has told you that what should be the ranking of the stability of this gene which we have. Right? So again, I think it is time we can break for few minutes and take up the questions.